So next biomolecule is proteins, and proteins are polyamides. Proteins are polyamides, means they have amide linkage. Amide linkage. They are also called as polypeptides. Polypeptides. That is, they have the peptide linkage. In peptide linkage, children, it is C double bond O and this NH. This is the amide linkage. CONH. It is amide linkage. Amide linkage or amide bond. Now, proteins are made up of basic components. Protein is also a polymer and is made up of amino acids. Proteins are made up of amino acid molecules. Amino acid molecules. Amino acid molecules, or simply we call alpha amino acids. Amino acid molecules or alpha amino acid molecules. Now, amino acid molecules, they have the character of acids and bases both. And this, here we have the H, here we have R, and this is COOH, this is NH2. So all the different bonds, all the different groups attached to the carbon are different. And this is very important part of the amino acid molecules, that they exist as Zweiter ion. They exist as Zweiter ion. How? This hydrogen, it goes to this, because nitrogen, it has the lone pair of electron. It has lone pair of electron. And as a result, what happens? This S3N, here it becomes plus, then C, H, this R, this C double O. This is called Zweiter ion. Zweiter ion. So, Zweiter ion is an ion having the negative and positive both the charges. So, this is very important point about the amino acids. Now, amino acids in large number, they join together to form the what? Molecule of protein. If we say here n moles of amino acids, and so this n moles of amino acids, they undergo condensation. There is the condensation between the different groups. So, condensation. So, here we have taken n moles, and as a result, here it is there. So, N, H, C, then H, then R, again C, here double bond O, then here this N of other molecule, then H, then here C, then H, R, then again here N, equal right here. So like this. Now this is the polyamide, because here we have the N. Thus proteins are polyamides or polypeptide. This is polypeptide linkage. And so there will be the loss of n moles of water. N moles of water is lost, and so this is the basic structure of proteins. Now let's see about the classification of amino acids. Amino acids they are classified into two ways. First is depending on their nature, and second is depending on their availability. Availability means in our body or in living system. So here first we'll see about the nature. So nature means acidic, basic or neutral nature. So first on this there are the three. First is acidic amino acids, then basic amino acids, and third is neutral amino acid. So amino acid is said to be acidic when it has more COOH group. Then basic amino acid, they have more amine group, and neutral equal COOH and NH2 group. When they are equal. So there are different types and we classify on this category. Now depending on their availability, and they are divided into two categories: essential amino acids and non-essential amino acids. So first is essential amino acids. So children, essential amino acids are the amino acids which are taken in form of food or dietary supplement or in the form of diet. So the amino acids which are taken in the form of food, we require proper amount of proteins in a balanced diet, and proteins take in different forms like milk, eggs, meat, and pulses. So they are all sources of amino acids. Now other is non-essential amino acids. So they are synthesized in the body of animals, not human. All the animals, proteins, all the living system, whether it's animal or plant, because all the seeds they are made up of proteins, and the nitrogen is very important factor. You are knowing from a small classes, nitrogen is key factor in the development of all the plants. Also the plant cells, they require some amount of nitrogen, and they take in form of nitrates. And why nitrates? Because all the plants, they produce seeds. All the seeds, they have cotyledons, and cotyledons, they are nothing but they are the source of proteins. So why it is so? We take different form of proteins because our body requires particular type of protein. We have different type of proteins in our body. We have nails, we have hair. So similarly in other animals also, like birds they have beak is also fibrous protein. Milk, it is globular protein. So this is a very important part. And so this non-essential amino acids, they are not essential. They are produced inside the body of organism by different biochemical reactions, catalyzed by some enzymes as well as by some bacteria. Now coming to a very important part, the structure. So children, proteins have 4 types of structures, and the 4 forms, they differ from each other in terms of the arrangement in terms of the arrangement of the protein chain, and first is called as primary structure. So primary structure is the native form. It is the native or original structure. Native means the inhabitant. Native or original structure in which a peptide molecule exists. The existing of peptide molecule, as they exist, how? Like we write NHCO. This N. So this is, this is numerous. So simple arrangement, what simply we can say? The representation of the polypeptide chain. This is what? This is polypeptide chain. So simply the representation of polypeptide chain is called as primary structure. Nothing. Just we write in a straight manner. Now second is the secondary structure. So secondary structure of protein means when the primary structure is folded at regular intervals. When primary structure is folded at regular intervals, there is the bending or the folding. The bending of this is the straight chain. Here is the bending. How? Like this. How is it present like this? Let it be. Then here C like this. Here also we have NH like this. Here they are held together. We can let this option be present here. It can be held together. Here there will be the hydrogen bond, or there can be sulfide linkage. So the very important part in secondary structure is that these folded structures, they are held together by different bonds like hydrogen bond, sulfide linkage, held different folds. They held different folds, and the secondary structure of the protein is intact. The weak bonds like hydrogen bonds, sulfide linkage, they are responsible for maintaining the secondary structure of proteins. And so this is the secondary structure. Now coming to tertiary structure. So structure is said to be tertiary when there is the holding of the secondary structure. When secondary structure of peptide chain or polypeptide chain is folded. So there is the holding of the polypeptide chain, and how it is like this. Just to take the case of this U, and how it is that inside this there is like this. So we draw like this. So the holding of the secondary, again there will be same bonds. Here we have the just white C double bond O N like this. Here there will be the N, here will be H. Then C, O like this. Similarly, here there will be CO. Here there will be NH. So this is the tertiary stage. Just the folding of the secondary on the each other. It is called as tertiary. Now coming to fourth, quaternary structure. So quaternary, as name indicates, four. So it is the basically haphazard arrangement or the irregular arrangement. It is the irregular distribution of protein chain or polypeptide chain. There is the irregular distribution. There is no uniformity. Like, there is total chaos and all the protein molecules, they have this type of, they are folded on each other, simply you can say. Here we say folded on each other without any specific pattern, and so this is called as tertiary structure. Now next very important part is denaturation. Denaturation of proteins. So denaturation is the loss in biological character. Loss in biological character. So it is the loss in the biological character. Biological character of a protein. Loss in biological character of a protein. The protein loses its biological character under some adverse conditions. There are the conditions which are adverse, like change in pH, temperature, growth of some microbe, etc. So the protein sample is no more biologically active, and only it can be digested by animals, or it is of no use. It loses its biological activity. And the primary, secondary, tertiary and quaternary, all the structures are lost. So polypeptide chain loses its identity completely. It loses its identity completely. All the structures, primary, secondary and tertiary, they are lost. And the polypeptide chain can not be reversed back. And only way is to break the chain into simple amino acids. Examples are curdling of milk. When milk is left unattended in warm weather, so milk starts curdling. So it is one example. Then boiling of eggs. When we boil eggs, when we prepare omelette, what happens? The liquid portion of the egg becomes solid. When we prepare chicken or cook meat, what happens? The flesh becomes hard because the flesh on heating, it loses its biological activity. Then, so there are different conditions. So, formation of cheese, formation of paneer, they are all very important. Now, children, last very important part is the functions of proteins. So, proteins are very much important for the animals. So, they are responsible for restoring and maintaining the damage. So, in maintenance and repairing of damaged part of body. Muscles, they are repaired. So, in growth and development of body. Growth and development. All the animals' body is made up of proteins, muscles. So it is there. So this is the very important part about the functions of protein. Now, next important part is globular proteins, and other is opposite part is fibrous proteins. So children, globular proteins are the proteins in which there are the globules, the molecules of the proteins, then form of small globules or balls. The molecules like milk, example milk, albumin, they are globular proteins. There are many more, insulin also there. Here, they have the fibers. Long fibers. Here we have the 6 molecules. So the molecules are small molecules, and they in the form of globules or small balls. But here the fibrous proteins, they are just stretched with thread. Example, here nail, horn, etc. And keratin is found. So fibrous proteins are water insoluble, and globular proteins are water soluble. So globular proteins are soft. They are soft, and they are hard. Like the hair, the nails, they are very hard. And this is due to the difference in their structure, difference in their bonding.